Hi, my name is Dr. Susmita Paul, and I am from the Assam Royal Global University, Guwahati, Assam. In this short video, I'm going to talk about plant growth promoting Rhizobacteria, its importance, and its role in agriculture. This particular topic is very much required in both UG and PG program in microbiology. To begin with, let us try to understand what basically are these plant growth promoting Rhizobacteria. As the name itself it suggests that these rhizobacteria, the bacterial form of microorganisms, are very much required in plant growth promotion, and hence the name. According to the presence of the microorganisms, which has been seen in the past few years, that these microorganisms are present almost everywhere. So we understand that these can be present in the plant as well. Also, in the vicinity of the plant root as well as its parts. Here in this case, we are talking about the soil bacteria, which has got the capability of colonizing the plant root. And these organisms are basically the plant growth promoting rhizobacteria. Now, these plant growth promoting rhizobacteria also help in facilitating the plant growth directly or indirectly. Of course, they has got their own mechanism of action, which could be direct or indirect. These help in decreasing the global dependence on the hazardous agricultural chemicals, as we understand that if microorganisms can be used instead of the chemicals, then in that case, we can depend on these kind of microorganisms instead of using the chemicals in the agricultural sector. These also help in stimulating the plant growth through mobilizing the nutrients in the soil, since we understand that these organisms are present in the vicinity of the soil. Now, let us try to understand what are the characteristics of PGPR or plant growth promoting rhizobacteria. These PGPR should be proficient to colonize the root surface, as we already have seen that one of the characteristics of PGPR is that it should colonize the root surface of a particular plant. This has got another characteristic that it should survive, multiply, and compete with other microbiota. That means there will be other kind of microorganisms which will be present in the environment. So these organisms, while present in the root surfaces or in the vicinity of the root surface, they have to survive. At the same time, they should multiply, and also they will be competing with these kind of organisms which are not the PGPRs. These also help in the plant growth promotion, and that is the most important characteristic feature of PGPR, that it should promote the plant growth. Otherwise, these will not be categorized under this particular category of plant growth promoting rhizobacteria. Depending on the functional activities, these are classified into four types. The first is biofertilizer, second, phytostimulator, third, rhizoremediators, and fourth, biopesticide. That means that PGPR can act as biofertilizer, as we have already seen that if these can be used as fertilizers in the fields, then in that case, the chemical fertilizers can be replaced. And also these have got the capability of acting as stimulators, hence called the phytostimulators. This can act as remediator, meaning it can help in degradation processes and hence called rhizoremediators. This can also act as pesticide because these will act on the pests and hence known as the biopesticides. So depending on their functional activities, these are categorized under these kind of um, types of PGPRs. Now let us try to see, depending on the presence of PGPR, they are categorized into two types, that is extracellular PGPR and intracellular PGPR, which is also known as ePGPR or iPGPR. So basically, when we talk about ePGPR, these inhabit the rhizosphere. or on the rhizoplane. Now, when we understand rhizosphere or rhizoplane, we mean that these are the area which is basically near the root surfaces. So hence, this EPGPR will inhabit the rhizosphere or the rhizoplane, or probably in the spaces between the cells of the root cortex. Whereas IPGPR will mainly inhabit inside the specialized nodular structure of the root cells, and hence the name says intracellular PGPR. To see some of the examples of extracellular PGPR, these could be Agrobacterium, Arthrobacter, Arvenia, etc., which are listed as you can see on the screen. To list some of the examples of intracellular PGPR or IPGPR, these could be Bradyrhizobium, Allorhizobium, and others which can be seen on the screen. So that is all in this short video. Thank you so much for listening.